In Chapter 50, we explore the incredible mechanisms behind sensory perception and movement. From detecting stimuli to muscle contraction, let's dive into how our bodies work in harmony to keep us moving. Stimuli are forms of energy detected by sensory receptors. Sensation occurs when this energy changes the receptor's membrane potential, triggering an action response that sends information to the nervous system. This input may prompt a reflex or more complex motor response, depending on the stimulus. For example, a mole in a tunnel eats if it finds food and keeps moving if it doesn't. Whether the input is present or absent, the information is integrated to guide the motor response. Sensory receptors detect stimuli and create receptor potentials. Changing the membrane potential, stronger stimuli cause stronger potentials, leading to more frequent action potentials in neurons or more neurotransmitter release in non-neural receptors. When the receptor is a neuron, it sends the signal directly to the CNS or central nervous system if it's a cell. It releases neurotransmitters to regulate the afferent neuron, which then sends the signal to the CNS. This image shows the difference between gentle or more pressure. Gentle pressure generates fewer action potentials, while more pressure leads to more frequent action potentials, activating more receptors based on the intensity. Muscle activity is a response that was inputted by the nervous system. The action of a muscle is always to contract. Muscle consists of bundles of fibers made up of individual cells, myofibrils, myosin, or thick filaments, aligned at the M line, while actin, thin filaments, attached to the Z lines, pointing toward the M line. Muscle contraction follows the sliding filament theory, where thin and thick filaments slide past each other. Myosin binds to actin, forming cross bridges and pulling thin filaments toward the sarcomer center. This process requires repeated binding and release, powered by ATP from glycolysis and aerobic respiration. So there's a relaxed muscle, and then you have a contracting muscle. The actin filaments are moving closer together, and then when you have reached full contraction, your sarcomer is much smaller because it has contracted as far as it can go. For this process, the myosin head on the thick filament uses ATP to reach its full high energy state. It then binds to specific sites on the thin filament or actin, forming a cross bridge. This pulls the actin filament toward the sarcomer center. Moving on to joints and muscle movement. With the presence of our joints, muscles, and skeletons, we are able to achieve locomotion. However, we must consider how these different components achieve locomotion. In this diagram, different types of skeletal systems transform muscle contraction into locomotion. Muscles are essential for generating force to enable locomotion. In complex skeletons, we can find three types of joints: pivot joints, hinge joints, and ball socket joints. Lastly, we have our different types of skeletal systems. The first and most basic type of skeleton is the hydrostatic skeleton, wherein it achieves the peristalsis locomotion as seen in the following diagram. For the second type, in exoskeletons, muscles attach to its interior, and when they contract, they pull against the exoskeleton, allowing movement. And lastly, for endoskeletons, muscles attach to its bones, enabling precise and powerful movements. They provide the framework for diverse locomotion strategies.